what I'd like to do with you today is give you some insight into some of the problems that we're working on now, or that I'm very interested in now, that have come out of 35 or 40 years of work using a small invertebrate nervous system. This is called the crustacean stomatogastric nervous system, and we use it. Over the years, I've used it to study many, many different things, and I won't have time to discuss most of them with you. But I just wanted you to see how it's possible to use a very well-defined nervous system to ask a wide variety of generally applicable problems, some of them which are as applicable to the lives of a lobster as to the lives of a human. But before getting going, I always like to acknowledge the people who've done the work. And my present lab is over there, including a few undergraduates. Most of them aren't on the slide, graduate students and postdocs. And then there's a large number of lab alumni, and then people who've gone on to industry and government jobs, including some of my ex-undergraduates, and people who now have faculty positions at a variety of other places or run their own labs. And then, of course, a number of eminent collaborators. And I like to show this in most of my talks because I believe it should give hope to all of those who are currently floundering in the purgatory, the postdoc purgatory. There are some of you, right? Yeah. Okay, so some of the problems that I'm going to be addressing with you are issues about how tightly tuned do the parameters that control synaptic strength and intrinsic excitability need to be for good enough behavior. And the other way of articulating this is to ask the question, how different are the brains of normal, healthy individuals? And if you think about it, I think everybody in this room knows that all of you have different brains. But as biologists, most of you were trained to do a series of measurements, calculate means and standard errors, because you always like the small error bars, and then to make the implicit assumption that all of the individuals whose data went into the mean are functionally identical. And I think it's only been in the last 10 years that we and other people are starting to unpack their data and really ask a number of very important and fundamental questions about how large a range of any one of the parameters in the brain can be and still have that brain or those circuits function properly. Then the other problem that I've become very preoccupied with in the last few years is a whole issue of what I'll call degenerate pathways or parallel pathways. The fact that most neurons in the brain are connected to other neurons in the brain by multiple pathways. And then we have the challenge of trying to understand how information flows through those different pathways. And then a problem I'm not really going to talk about with you today because those are the slides that I dumped. You know, I have to make these really painful decisions. But it was really very painful last night to say, am I going to do this or this? Anyway, is a series of sort of computational and experimental studies we've been doing to try and understand what kinds of control systems could be used to deal with the issue that while brains are alive for 100 years, neurons are alive for 100 years, ion channels, receptors, and synapses are turning over all the proteins that are important for synaptic transmission and excitability, turn over in hours, days, and weeks. And I think this is a very, very fundamental problem that we've been working on for the last 25 years, and we continue to try and work on along with many other people. Okay, so the system we study is the crustacean stomatogastric nervous system. The nervous system comes from the stomach of a lobster or crab. What we do is we get the animals from the fishermen down on the wharves in Boston. Actually, the animals take a taxi to the lab because we don't go get them anymore. They come to us. What? No, it's actually a delivery service. They're not so good at dialing phones. And then we hold them for a while. We acclimate them to constant temperatures, and then we dissect the stomatic-gastric nervous system from the surface of the stomach. Now, most of you don't know that lobsters and crabs have a very complicated mechanical structure as a stomach. It's completely different from a vertebrate system. It's not an autonomic nervous system. Rather, the muscles and the ossicles are moved because of motor neurons that fire in rhythmic discharges. So it's much more like your mouth or the respiratory center in you than it is like a stomach. And what you see here is how we do the experiments. This is a full-size Petri dish. The stomatic-gastric ganglion is in the middle of it. We desheath it. We record from 2, 3, 4, 5 sharp microelectrodes from the somata of the neurons. Most of the neurons of the stomatic-gastric ganglion are motor neurons, and that is why the system has been so good, because you can record from the motor neurons, but the pattern-generating circuitry that gives rise to these rhythms is actually at the level of the motor neurons. We leave attached the motor nerves that would go to the muscles that are now several flights down, and we put extracellular recording electrodes on the motor nerves to look at the discharge patterns. And then we often leave attached 3 anterior ganglia that send descending modulatory inputs into the stomatic-gastric nervous system. So if you do this, you can see here simultaneous intracellular recordings from 3 of the neurons in the what we call the pyloric rhythm. Here's LP, PY, PD. LP, PY, PD. This is a triphasic motor pattern. It's ongoing in the animal always. Like your respiratory centers, it's respiratory rhythms, it's always on. Although, like your respiratory rhythms, it can be modulated in frequency and phase relationships according to needs of the animal. Down here is an extracellular recording showing these same 3 units, color-coded with LP, PY, PD, LP, PY, PD. LP is a constrictor neuron. It's part of constricting a valve at the back end of the pylorus. PY is a second constrictor, and PD is a dilator neuron. So you have constrictor 1, constrictor 2, dilator, constrictor 1, constrictor 2, dilator. Now, this is a connectivity diagram. Today would be called a connectome that describes, ooh, you guys awake? People often laugh with that. Okay. That describes the circuitry that gives rise to this motor pattern. Now, I'll just very, very quickly talk you through it. The anterior burster is an interneuron that is an endogenous oscillator as a first approximation, and it's electrically coupled to the 2 PD neurons. So in the crab, there are 26 neurons in the stomatic-gastric ganglion. There's 1 AB, 2 PDs, and in all of my circuit diagrams, the resistor symbol means electrical coupling. And then in all my circuit diagrams, all of these synaptic connections with filled circles like this are chemical inhibitory synapses. So the way this works is the oscillator here drives this oscillation. So the PD depolarizes, fires a burst of action potentials, and then does that repeatedly. The AB and the PD neurons both rhythmically inhibit the LP and the PY neurons, and they fire on rebound from inhibition. So the rhythm exists because of an intrinsic oscillator, and then the timing comes about because of rebound from inhibition. And rebound from inhibition here is a very important timing cue, and this is often the case in locomotory systems or sometimes in the brain as well. Now, if I had shown you this connectivity diagram, as we used to call it, or wiring diagram, which we've had since 1980 or so, and you didn't know what this looked like, there's absolutely no way on the basis of this connectome that you'd be able to predict these dynamics. And that's because the information that's missing here is the nature, all the voltage and time dependent currents in each of the cells, and the strength and time course of all the synaptic inputs. Without all of that information, you wouldn't be able to go from the connectome to the dynamics. And I think that's a really important warning for those of you who are trying to study complicated brains. A connectome that we, and I think the C. elegans people as well, would say the connectome is absolutely necessary and completely insufficient to understand how the brain does anything. Okay, so what have we been doing since 1980, you might ask? What we've been doing since 1980 is asking a whole series of questions about how does one really understand dynamics in terms of the underlying components, and how does one understand modulation of those dynamics, how does one understand the robustness of those dynamics, and how does one understand how an animal can continue doing this for 7 years before we catch him in the waters of the North Atlantic. Now, just to make things a little more interesting, I'd like to show you the other thing that the stomatogastric ganglion does. You saw before the pyloric rhythm, and this is a now much slower time base. So the pyloric rhythm here on the fast time base is up here, LP, PY, PD. There's also a gastric mill rhythm. And there are 3 teeth inside the stomach, and you can see that with the depolarization of the LG, a burst of action potentials. And then you can see LG fires in time with GM and in alternation with DG. And I'm showing you this slide only because I want you to also see there are 2 neurons here called IC and VD, and they fire in an envelope in time with the gastric mill rhythm, but they're also firing in phase with the fast pyloric rhythm. So these neurons, these 2 neurons are, if you will, all multiplexing or participating in the generation of both rhythms on 2 different time scales. Now, I'm only showing you this to set the stage for the first take-home lesson I'd like to make. This is now the connectivity or the connectome of the full stomatogastric ganglion, where the neurons I showed you before are over here, and the gastric mill neurons are over here. Now, I've had a series of really wonderful students and postdocs in the lab who have all come to me saying they want to build a model of the SDG, and I always say yes. And then I sort of start asking how long is it going to take them before they decide that's too hard. And it usually takes, if they're really smart, it takes 2 days. If they're a little more of a novice, it takes them 3 or 4 days. And if they're really clueless, it takes them 3 weeks. But they eventually all decide that's too hard and come up with something else to do. Well, Gabrielle Gutierrez said that to me, and then 2 days later she came and said that's too hard. But I noticed something. And she noticed something that we had never noticed, which is at the core of the network, there's a 5-cell symmetrical circuit. So you see here, PD and LP are reciprocally inhibitory. LG and interneuron 1 are reciprocally inhibitory. PD has a monosynaptic connection with IC. Interneuron 1 has a monosynaptic connection to IC. So PD has a monosynaptic connection to IC and a polysynaptic connection through this electrical coupling. IT1 also has a monosynaptic and the polysynaptic connection. And Gabrielle started her life out as a physicist, and so usually as a physicist or a mathematician who'll notice these pieces of symmetry. But the reason why Gabrielle was interested in this is because reciprocal inhibition is a circuit motif that's very commonly found in many systems, but it usually produces, reciprocal inhibition usually produces alternation. So this is basically a simple 2-cell circuit that can be called a fast oscillator. LG and interneuron 1 is a slow oscillator, again, is reciprocal inhibition. And IC is one of those neurons that can switch between the fast and slow rhythms or can participate in both. And so Gabrielle looked at this and she said the problem she really wanted to study was what features of the circuit or what parameters would govern whether a hub neuron participated with the fast rhythm or with the slow rhythm. So now we're going to leave the details of the stomatogastric ganglion, just say it created an incentive to build this 5-cell circuit and to use it as a way of understanding what factors would control whether IC fired with the fast rhythm or slow rhythm or else. So Gabrielle decided to model all the neurons as oscillators. So these are her model neurons in isolation. And she now renamed them. She called one neuron F1, that's fast 1, fast 2. HN is the hub neuron, S2, S1, slow 2, slow 1. And you can see this one is a fast oscillator, this is a slow oscillator. That's an oscillator with an intermediate frequency. And when she hooked them up, here's that fast oscillatory circuit. You can see F1 and F2 are firing in alternation. S1 and S2 are also firing in alternation when she hooked them up. And if she hooked up the core through these electrical junctions, as you might expect, they all synchronize. So now we can ask, what happens when you hook them all up? So here's the full circuit. And in the next few slides, we're going to use a few little nomenclatures. The synapses from S1 to HN, or F1 to HN, Gabrielle is calling G syn A. That's the conductance of synapse A. These synapses she called synapse B, so we have G, that's the conductance of G syn B, and GL is the conductance of the electrical synapses. So what Gabrielle wanted, and this was actually very clever, she wanted a way of displaying the frequencies of all the 5 cells in the circuit simultaneously. So she came up with this method of displaying them with 5 concentric circles and a square. So F1 is on the outside, F2, HN is the square, then S2 and S1, and they're on a heat map. So this would be fast, medium, and slow. So now we can look at the behavior of all the cells and classify the behavior using this device. So one set of circuit parameters would give F1 and F2 are firing quickly. The HN is firing with the slow rhythm, and you can see that plotted here. Another set of parameters, they're all 5 of them, are firing at the same frequency. You can see that here, and that's why they're all green. And I don't know if any of you noticed, but for you, I wore my blouse. You see what this is? You'll see in a moment. So I walked into my office one day, and I found this on my desk, and I knew it had come from Gabrielle, and so I walked when she eventually came in. I said, "What's this?" And she said, "Well, that's a parameter scape." And I said, "I want a blouse." And she said, "I'll make you a blouse for my thesis defense." And I said, "Fine, you have a deal." So she did. So I thought I'd wear it for you. So what she's done here is she's plotted the conductance of G syn A on the x-axis and the conductance of G electrical, or GL, on the y-axis. And now you can see the boundaries of the circuit behaviors as a function of these 2 synapses, where everybody's plotted like this. So you can see up here there's a big part of parameter space, which has F1 is firing fast and the other 4 neurons are firing slowly. And as you go just a little bit to the left of that, you have a piece of parameter space where you have 4 of the cells firing fast and only 1 of them firing slowly, right? And then here you have a whole region where everybody's firing at the same frequency, and that's somewhere under my arm, and et cetera. There are all these different dynamics. And so this tells you, this is a really easy way of visualizing how the changes in the parameters can influence the circuit dynamics of the whole network. Now I'm just going to make 2 points with this. Point 1 is if you now have a modulator, a modulatory substance which influences G syn A. If you start out over here, a small effect of modulating GCN can actually, G syn A, can take you from showing this behavior to showing this behavior by going through actually another bifurcation there. So a small amount of modulation if you start out over here, can produce a massive change in circuit dynamics. On the other hand, if your starting state were over here, then you could have a much bigger change in synaptic strength, but produce no essential change in the qualitative behavior of the circuit. So this tells you, it sort of visualizes for something you've always known, which is that the effect of a modulator, the effect of a perturbation, will often depend on the starting state of the network. But this is just a really easy way of seeing it. And I like to dramatize this because I think it's a really good reminder that it's not how big the effect is, it's where the circuit is before that effect takes place. Now, I'd just like to show you 2 more slides, and this next one is why I really wanted to show you this work. And this is what we call degenerate mechanisms produce the same change in behavior. So here's Gabrielle's starting condition with F1 and F2 firing fast, HN is firing slowly with the slow neurons. And then she did the manipulation of decreasing G syn A, as you can see here, and HN flipped into firing with the fast rhythm. Then she went back to her starting condition, and she decreased the strength of the electrical synapse, and she flipped G-syn A into the fast, she flipped HN into the fast rhythm. And then she did a different manipulation. She decreased G-syn B. And again, she was able to flip HN from being slow to being fast. And the reason I'm showing you this is because these are 3 entirely different cellular mechanisms that are producing the same phenotypic change. Now imagine you're a graduate student here at Dartmouth doing an optogenetic experiment to manipulate, or a pharmacological experiment, or a genetic deletion, or whatever you do to influence G-syn A, and you get this change in phenotype, and you write a paper, and you send it to Cell, and they publish it, and your paper says, oh, it's G-syn A that's controlling the behavior of the circuit. And then some poor graduate student sitting at, oh, I don't know, it's Stanford for Rob, does this experiment, and they see this, and they say, oh, that person at Dartmouth was crazy. They got the wrong answer. And then there's some really sad student sitting in Berlin who does this experiment and gets the same answer. And by this point, the paper can only be published in, I don't know, +1, because these guys have already gotten it half right, right? Third right. Anyway, the answer is that all 3 of these mechanisms can produce the same, what looks like the same change in behavior. And this tells you this occurs because of what I'll call parallel pathways. That's because there are multiple paths by which F1 and S1 can influence the HN cell. Now, this is a 5-cell circuit. Imagine what's going on in the brain. Everything in the brain is connected to parallel pathways. So this tells you that when you do a manipulation and you see a change in circuit behavior, or circuit behavior in animals' behavior, the most you can say is, I may have stumbled across a mechanism that could be responsible under certain conditions for producing that change in the behavior, but you have not ever, you can't make the claim that you found the mechanism that produces change in behavior. Unless you know the connectome, and in detail, you won't even know where the parallel pathways are. And so that's why knowing the connectome is so important, because if you know the connectome, you can then see where these parallel pathways might be, and then you can go about testing them all. But in the vertebrate brain, that's a little bit hard. Okay, I'd like to show you one more slide from this work. And in the previous work, what Gabrielle was doing was changing the strength of synapses. And here she wanted to look at changing the intrinsic properties. So she took the HN neuron and played with the calcium and the potassium currents that give it its oscillatory behavior. And here you can see it still has the same frequency in these 4 cases, but it has a very different waveform. And then she embedded each of these 4 neurons into 2 different circuits. Circuit 1 had a strong synapse from F1 to HN, and circuit 2 had a very weak synapse from F1 to HN and a very strong electrical synapses. And now here you can see what happened. So here is HN in its different incarnations in this circuit, and you can see the circuit is mostly unchanged, although the HN cell changed its behavior because of the effects of the modulator. Nobody else in the circuit cares. On the other hand, when the circuits start out in this condition, now when she puts those same HN neurons or does that same modulation of the HN's intrinsic properties, you can see the whole circuit changes its activity. So it goes from 2 fast and 3 slow to everybody firing the same frequency to here to one of the cells actually even dropping out. So this tells you that depending on the starting condition of the circuit, modulating a single neuron can either influence only that neuron, or the effects of modulating one neuron can percolate much more widely and influence many other cells in the circuit. And again, this is something that's really important to remember, because all that's different between these 2 cases is what the starting sets of parameters were. Now I'd like to just summarize that and just say degenerate circuit mechanisms help ensure that, well, I didn't really say that, but the important circuit functions are robust, and different underlying circuit parameters can account for state dependence of modulator action. Now, I'd like to move on to another study which was done before this, which deals with other features of what we'll call state-dependent or multiple solutions, but deals with some of the same issues and some different issues. Astrid Prince was a postdoc in the lab, and she's now on the faculty in Emory and has a very fine lab of her own. And she wanted to build a pyloric network model. And so she wanted to start with some models we had in the lab, and she wanted a good model for the AB and the PD cell, the pacemaker kernel, and a good LP model and a good PY model. So she started trying to fine-tune neurons to get model neurons that behaved like she thought they should. And it was very hard because fine-tuning a conductance-based model is always very hard if you try and do it by hand. So eventually she gave up with that, and she took a model neuron that had 8 currents, and she chose 6 values of each of the 8 currents, and she simulated all against all. So she generated a database of 1.7 million model neurons, and then she selected from those neurons that she thought would be good representations of each of the neurons in the network. And then she did the whole thing again. She chose either 5 or 6 values for each of the synaptic currents. And then she simulated all against all. So she simulated 20 million model networks. And then she classified their behavior. And what she saw was, if she started out with 20 million, 19% of them were triphasic in the right order, but only 2.4% of them were triphasic in the right order, but had attributes of what we call the pyloric rhythm. That is to say, they were triphasic in the right order, and they matched data that Dirk Bucher had collected from 99 animals. And that is to say, his data had a range of frequencies, and a range of phase relationships, and a range of the number of spikes per burst. And then Astrid was able to find all those networks that fell within those ranges. So this at the time was a real paradigm shift, because instead of trying to build a single model that was representative of the mean of a collection of data, what Astrid was trying to do was find a family of models that represented the diversity in the biological population. Because that then allows you to start asking, well, what parts of these models are important or may be correlated to produce those behaviors. So when we were writing this paper, Astrid brought me these two, or I said to her, find me 2 networks that produce very similar behavior but have different underlying conductances, because I thought that was the most interesting part of the paper. So this was model circuit 1, model circuit number 2. And here are many of the parameters that give rise to these behaviors. So these are some of the membrane conductances, and these are some of the synaptic conductances. And the details aren't important, but it's clear that if you just see that this set of parameters and this set of parameters are entirely different, and yet together they're producing very similar behavior. So this was a very important paper at the time, because it really said to the biological community that individual animals might really have quite differing sets of parameters that would give rise to good enough or healthy behaviors. And you can see that here the H current IH and PY is big, here it's small. The PY-LP synapse is big here, here it's small. AB-LP synapse is small here, here it's big, et cetera, et cetera. So they're very different. So faced with this, we wanted to ask, in biological literature, how variable the parameters in real biological networks, and do perturbations reveal cryptic animal-to-animal differences? And then how reliably can animals with intrinsic variability with multiple solutions respond to perturbations? So I'm probably going to skip most of this. We spent a lot of time actually doing voltage clamp measurements to measure the maximal conductances of individual ion channels in the same neuron across animals. We then took some of those same neurons and did single-cell PCR and measured the mRNA for a bunch of different ion channel genes. And since then, Dave Schultz and he was one of my postdocs who recently got tenure at the University of Missouri. We've looked at the distribution of 80 or 100 different ion channel genes in every single identified neuron in the STG. And you see across the population 3 to 6-fold or 2 to 6-fold ranges in the mRNA expression or in the strength of any of the synapses that we've measured. And that is despite the fact, as you see here, if you just record the motor patterns, they look very, very similar. But on the other hand, if you look at any of the individual parameters that you could measure biologically, they'll have this range in them. So, and I'm going to skip all that because what I wanted to do is spend a little bit more time talking about the more interesting set of experiments, which are to ask, if you've got 2 animals or 10 animals, and they have a different set of solutions, one of them has a big K current over here, one of them has a small K current and a big calcium current, this one has a big H current, and that one has a big glutamate inhibitory synapse, and that was a smaller one, but a stronger electrical synapse. Can you expect that, 100 crabs with that range of conductance densities and synaptic strengths, if they're sitting in their world, can they nonetheless be healthy and robust in response to the perturbations that they need to see? And this came up when I first started talking about this, and theorists would say to me, well, if you have 2 animals with very different sets of underlying conductances, yes, they may look similar until they're perturbed, but they're bound to behave differently when perturbed. And so then we thought we would have to do this kind of experiment and take a whole bunch of animals, which we already knew had different sets of underlying conductances, and then perturb them and see how robustly they could respond to perturbations. And obviously the perturbations that you want to look at are the perturbations that the animals have to deal with. So if you're living in our neck of the woods, not maybe so much your neck of the woods, you're not so close to the ocean, but in my neck of the woods, the perturbation that these crabs that live right outside my door have to deal with is, among other things, they have to deal with temperature changes in salinity, et cetera. So, but temperature is a global perturbation that will influence the behavior of every single biochemical molecular process in a cell. And so now the question is, can you build, or are these networks temperature compensated? So we often talk about a Q10, which just characterizes the effects of temperature on a biological process. A Q10 of 1 is a process that's completely temperature compensated. There's an exponential in here, so Q10 of 2 already blows up. And a Q10 of 3, you can see, changes a lot with temperature. Now, ion channels and most biological systems have Q10s between, rate constants between 2 and 4, but there are some rate constants in some ion channel genes that can go up to 50 or 100. So we didn't exactly know what to expect. But we figured out very early on that if you had a bursting neuron and each ion channel had a different set of Q10s, and the way the burster worked with the inward and outward currents, it would be very easy for that to become decompensated. So, and just to show you that, because the rate constants would all get out of whack with each other, if you start out with a bursting neuron that you see here, and you change temperature, and these are 3 different bursting neurons that all look the same, this is what those theorists were saying to me. You start out, they all look fine at 10 degrees, and now you increase the temperature. This one starts behaving tonically and then crashes. This one becomes a one-spike burst and then crashes, and then these go through other behaviors as you go cold. So it turns out it's actually very hard to find neurons that will maintain the same basic property over a big temperature swing. However, Tim O'Leary, and I'm skipping skipping a lot to tell you this, had a principled way of finding neurons like this. And so here are now 1, 2, 3, 4, 5 neurons that are bursters that he found looked by using a homeostatic rule, and these neurons all can maintain their bursting properties as you change temperature because they have the right set of matched rates of turnover of their channel genes. But now what about the biology? So Lamont Tang did the first set of experiments. Here's a rhythm at 7 degrees. Here's one at 19 degrees. Looks the same, only this one's faster. And the rhythm has an apparent Q10 of about 2.3. This is a semi-log plot. And you can see it goes faster, the rhythm goes faster as the temperature increases, just what you'd expect. What I was really surprised about when Lamont brought these data is here you can see that the phase relationships, that is to say the relative timing of all the neurons in the rhythm, were perfectly maintained as the temperature was changed. So this network has a way of maintaining the right timing while the frequency is changing over a very large frequency range. And this is actually not an obvious thing to do, and it took us a while to figure out how it does it, and I won't have time to really tell you about that. But I will show you the next thing is, which Lamont did discover, that after you went through this large permissive temperature range, which was basically from about 6 degrees to something around 25 or 26 degrees, if you push the temperature still higher, you'll get what he called crashes, which is a loss of the rhythm. And then if you bring the temperature back down, the rhythm comes back. So you have a large range of temperatures in which the animals manage just fine, and then the rhythm crashes. And then the next question was, if you have different animals with different underlying conductances, how am I doing? He's making this up. With different underlying conductances, you might expect them to show different crash behaviors. You might expect the change in temperature to reveal those different underlying differences. So what Lamont did to show that is he plotted spectrograms now in 5 different animals, and I'll just ask you where you look at the power from 0 to 6 degrees, PYLPPD, PYLPPD, and here they're all behaving well, but by the time you get to 27 degrees, this one's showing all sorts of intermittent disrupted behavior, this one a little bit of disrupted behavior. These still look okay, and by 31 degrees, they're all disrupted, but they're all disrupted differently. So the question then was, and I think we seem to think that we were seeing evidence that these different underlying parameters were producing crashes by different mechanisms. So the next question, I'll do this very quickly, I promise, was to say, could you change that by influencing the modulatory environment? And so Sarah Haddad, who just finished her PhD, so this just summarized what I said. Temperature crashes are widely variable because of the variability, but now can the modulatory environment alter crash susceptibility? And I'm going to have to skip all this stuff, this beautiful stuff. And just to show you, it's in the wrong order, to show you that Lamont's data had all been done with these modulatory inputs left intact, with a starting case like this. What Sarah did is she removed the descending intrinsic modulators, and you end up with a very slow or irregular rhythm under those conditions. And then she wanted to add back some of the modulators. So here I'm just showing you 3 animals, animal 1, animal 2, animal 3, with modulators all removed. And you can see they're quite variable to begin with. And then as you increase temperature, this one maintains a pyloric rhythm, and this one becomes completely disrupted. And Sarah then wanted to classify the kinds of disruptions or patterns that she saw. So she came up with a way of classifying the rhythms, either a normal triphasic rhythm or a slow triphasic rhythm or a silent rhythm or just the PDs or intermittent, and then she's color coding them. And then what she's done is she's, oh no, I lost a slide. It's okay. You don't need to see it. And then, using that same classification, she did the same experiment with two modulators, oxytremorin and proctolin, where now you're seeing it, the two animals in oxytremorin and two in proctolin, and you can see what dynamics they show. And here, now color-coded, you can see these guys are very, very regular. And the slide that I lost showed all sorts of disruptions. But now we do the same thing in serotonin. And so here are 5 animals in serotonin, 1, 2, 3, 4, 5. And as we change frequency, you can see they become more and more disorganized. And now, if we look in serotonin, you can see that every one of them has different sets of color blocks and makes different kinds of transitions. So what the oxytremorin and the proctolin did is they stabilized the rhythm, and the slide that was missing just showed you in the absence of the modulator. And serotonin made the rhythm more sensitive to temperature. Now, serotonin acts on a bunch of different voltage and time-dependent currents, and IMI activates, the other two modulators activate a current we call IMI, which is like the NMDA receptor current, and it acts to stabilize oscillations. So the take-home lesson from this is, and then I'll be done, is the effects of modulators on network stability. Proctolin and oxytremorin activate the voltage-dependent current that tends to enhance oscillator function, and they stabilize the rhythm against the temperature perturbation. And serotonin acts on numerous currents and destabilizes the rhythm. And this just summarizes what I just told you, I think. Yes. And I'll just end here by just reminding you that at low temperatures, all networks are well behaved, showing consistent changes in frequency and outstanding temperature compensation, despite the fact that they have this underlying parameter differences. They all crash differently with different dynamics, and some modulators expand the stable range of temperatures while others make the preparations more temperature sensitive. And I'm very sorry for going long. Not too long.
Okay, I think we probably do have time for a question if maybe as our next speaker gets ready.
Yeah.
So very nice that there's a wide range of possible outcomes in the variable sense, given where the circuit started from responsive perturbations. Do you know if there's any sense of whether circuits that for them it's most important to be robust use less of their parameter space, whereas for circuits where maybe it may be more important to be flexible, use larger amounts of the parameter space? So for
it's actually not necessarily. I think you made a leap of faith.
Yeah, I'm just curious if there's any.
It's not clear that what you said is right.
Yeah, I guess that might
okay.
Be, is there an evidence that
so,
you may imagine breathing you want to use less of that than you use parameter space. Maybe association or you want to use larger swaths of parameter space so that they may have
this is a rhythm when I'm studying, which is important for feeding in the animals. So there's nothing more important than feeding. My suspicion is that these numbers and these degrees of freedom are probably pretty general. I have a personal feeling that getting things tuned within 2 and 6-fold is something that biological systems, homeostatic mechanisms can do. Getting things tuned within 2% is too hard. Biological systems probably can't do that. And once you go beyond a certain point, then you're wandering all over the world. So I should say that even though there are multiple solutions to being a good crab, not all solutions work. right? Remember, in that 20 million networks, only 2.3% of them actually produce the right behavior. So the successful solutions can be quite different from each other, but they're not random in the solution space. They come with certain very important sets of correlations. I don't know if that helps you think about this. In terms of robustness, I think all parts of the brain need to be robust.